life is actually a disequilibrium. If you want to be one with the universe, it's simple. Just die. I can explain. So what does a simple soap bubble have to do with life? Well, a simple soap bubble has a lot of similarities with the cell, and the cell is the basis of life. Wherever there's life, there's a cell around, a biological cell around. And so we talk about the similarities between the cell and a soap bubble, and we will get some inferences on life. Distinct barrier, inside, outside, detergent barrier, as I've said before, okay? Much like a regular cell. There's air trapped on the inside, so that makes asymmetric environment. It's a disequilibrium. If we want to make the inside now the outside, it's a really simple procedure. To make it equilibrium, all we have to do is pop the cell. So now there is no inside or outside, and the cell is gone. To actually see a living biological cell, we need a light microscope. Of course, we don't have that right now, so instead I'm going to use a prop, okay? This giant hamster ball. You can see that it's got a membrane-bound nucleus in it. That's the white balloon. So this would be considered a eukaryotic cell, which is much like the cells that make up our body. Also inside are some other components. These specifically are to represent the other cellular organelles. Many of those are membrane-bound. You can see right now it's pretty confusing. So to do research, you need to make things a little less chaotic. Now, an important feature of biological research, or research of any kind, is to make things more simple or less confusing. And to determine what's the inside or the outside of the cell or the organelle, there's actually a relatively simple way of doing that. I have a little pointer here, okay, and my hamster ball here. I've removed these ends just so that for demonstration purposes only, so imagine it's still part of the cell membrane. Okay? But the pink end is on the outside now, and if you push it through the membrane, it's now on the inside of the cell. Outside, inside, outside, inside. If we push it all the way through the next membrane on the other side, we've gone from outside to inside to outside. Okay? You'll notice something really interesting, is that if you go from outside, inside to outside, you've crossed two membranes. But if you go from outside to inside, you've only crossed one membrane. And so when you're dealing with the internal organelles, you can actually do a similar thing. Is all you have to do is count how many times you've crossed a membrane. If it's an odd number, you've gone from the outside to the inside. If it's an even number, you've gone from the outside to the outside. So hopefully my little demonstration has shown you that there's a distinct outside and inside to the cell, and on the cellular organelles there's also a distinct outside and inside to the cells. If that wasn't clear, I'm putting up a diagram of a cell, a cross-section of a cell, to explain it a little more fully. This is a drawing of the cross-section of a eukaryotic animal cell. It is easily seen here, the various membrane-bound organelles that I was speaking of. And in fact, there are various labels here too. But what I wanted to show you from this diagram is there on the red arrow on the image right, okay, where it says out and in. You can see that that red arrow, when it crosses the cytoplasmic membrane, that's that surrounding area on the outside, when it crosses that membrane once, it goes from the outside of the cell to the inside of the cell, into the cytoplasm. This next slide is here to show you how
important it is to determine what's the inside of one of these cellular organelles. If you look down to image right, that red arrow is pointing to a yellow area, which is actually the lumen, or the inside of something referred to as the endoplasmic reticulum. So is that the inside or the outside of the cytoplasm or the cell? Well, we can do this with the arrow. You look and see, the arrow crosses the cytoplasmic membrane once, and then it crosses the endoplasmic reticulum once. So remember what I said, if it crosses an even number of times, it's actually going from the outside of the cell to the outside of the cell. So technically the endoplasmic reticulum lumen is the outside of the cell. Now in this third figure, just as a test, I've moved the arrow, the red arrow, just up a little bit, so you see it's actually pointing to the white and not the yellow. So it's in the cytoplasm, pointing to the cytoplasm. So technically, remember, the cytoplasm is the inside of the cell. But let's do the membrane test. Let's count the membranes. So we got the cytoplasmic membrane, that's one. We've got that one endoplasmic reticulum membrane, that's two. And then we go across the lumen of the endoplasmic reticulum through that other endoplasmic reticular membrane, and we end up basically back on into the cytoplasm. And so remember, again, odd numbers, we're going from the outside to the inside. So see, that worked out. Okay, so the arrow is pointing to the inside or the cytoplasm of the cell. Now on this fourth diagram, I want to make certain that you see the importance of this aspect of determining what's the inside of the outside of the cell. You see where the arrow is pointing to this little vesicle that's fused to the cytoplasmic membrane. That vesicle actually came off of the end of the endoplasmic reticulum, and that's the way secreted material makes its way out. So it gets formed in the endoplasmic reticulum. The endoplasmic reticulum makes a membrane-bound vesicle that goes into the cytoplasm, and then it moves out to the cytoplasmic membrane, and then it physically fuses with that membrane. But as you can see, when it fuses, the membrane just opens up and it dumps right into the outside of the cell. So the lumen of the ER, which was technically the outside of the cell, is shown to actually dump into the outside of the cell.
So hopefully from that diagram and what I've said already, you've got a clear idea that one of the cell's major functions is to keep the outside out and the inside in, and that there's a simple way of distinguishing what's the outside and what's the inside of the cell. So this difference between the outside and the inside means there's asymmetry, so it fits with the conclusion that life is a disequilibrium.